All right, welcome everybody to the January GOA-ON Webinar, the first of the year. I am Katarina Scholl and I am an Associate Project Officer for Ocean Acidification at the Intergovernmental Oceanographic Commission of UNESCO. I'm based in Paris. And I would like to welcome you to this webinar. So the GOA-ON webinar has 4 sponsoring organizations. First, obviously, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the National Oceanic and Atmospheric Administration of the United States. Third, the IAEA OAICC, which is the International Atomic Energy Agency Ocean Acidification International Coordination Center, and the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. For those of you who are new to GOA-ON, it's a collaborative international network of over 900 members now from 105 countries, and it's designed to detect and understand the drivers of ocean acidification and the resulting impacts on marine ecosystems. During this presentation, all participants are in listen-only mode. You're welcome to type any questions into the question box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring all incoming questions and we'll pose them to our speakers during the question and answer section, which will begin immediately after the presentation. Today's webinar is moderated by Dr. Nina Bednarczyk. She is a prominent ocean acidification researcher and also a member of the GOA-ON OA Mediterranean Hub Steering Committee. Nina is a senior scientist at the National Institute of Biology in Slovenia. Nina, welcome. Thank you for doing this today. And yes, I'm turning it to you now.
Great, thanks so much. Thanks so much. And it's a pleasure to be hosting this important and very interesting session. I think this is the future of trying to start establishing the impact of multiple stressors, in this particular case, warming and OA on the Mediterranean basin. And just for a little bit of a background, the Mediterranean Sea is a hotspot of global change due to its semi-enclosed configuration. Mediterranean sea surface water is expected to be 20% above the global mean projections. And as for warming, ocean acidification seems to be more intense than the global ocean. So both the warming and OA are more intense in the Mediterranean, or getting more intense rate-wise than the rest of the global ocean. And so throughout this webinar, we'll present the impact of combined OA and warming on some emblematic Mediterranean calcifying species. So we have 2 speakers here today with us. Chloe Carbonne, she's going to be our first speaker, and then Maximiliano Szkope. So maybe I can introduce the second speaker a little bit later, just before his talk. And so I'll start with Chloe first. So Chloe just finished her PhD at the Lab of Oceanography of Villefranche, Sorbonne University in France. And she has focused her research on the physiological impact of global change, in particular warming and acidification, on different cnidarians, with particular interest in corals. During her PhD, and this was done under the 4Ocean and MOPGA project, she combined in situ monitoring with scuba diving at naturally acidified sites in Ischia, in Italy, and with the long-term laboratory experiments with controlled temperature and pH. So Chloe is currently looking for a postdoc position, but the work she's presenting today is assessing the impact and tolerance of low pH on different life stages of 2 endemic coral species, Astroides calicularis and Cladocera cespitosa, both living in CO2 vent sites at naturally acidified environments. All right, Chloe, the floor is yours, please.
Thank you very much. Just have to share the screen first. Can you see my screen right now?
Perfect.
Yes, okay. Well, thank you, Nina, for the presentations. I just have finished my PhD at Sorbonne University, and I will right now talk about some of the results of the experiment of my thesis, which leads to the conclusion that early life stages of Mediterranean corals are more sensitive to ocean acidification than adults. I will only speak about Asteroides calicularis and not Cladocora. Our study sites are in Ischia, a little island in Italy, where we can find CO2 vent sites of pure CO2 bubbles of volcanic origin that naturally acidify the seawater around 7.8 of pH. This naturally acidified environment can be used as natural laboratories to study the future impact of ocean acidification, as they present a pH close to the one projected for the end of the century. In the acidified sites and in 2 ambient pH sites of Ischia, we can find natural population of the azoxanthellate or non-symbiotic coral Asteroides calicularis, endemic to the Mediterranean Sea. During my PhD, I had the chance to work on the full life cycle of this species, which is gonochoric, which means that it has sex-separated colonies, and which is also an internal brooder. The fertilization occurs inside the polyps of females, and the larvae will be released by the female polyp when it's mature. Acidification usually have an impact on calcifying species, mostly by disrupting calcification. However, as corals are able to live in the acidified system in Ischia, we wondered if the colonies in the vents were more tolerant to low pH than the one from ambient sites. To test this, we sampled colonies from the CO2 vent sites and an ambient pH site, and we exposed them for 6 months under an ambient pH and a low pH of 7.7, which is the projection at the end of the century under the RCP 8.5. Here in the reddish colors, we have the colonies from the vent and the blue from the ambient site, and I will keep with this color for ambient and vent sites. And no significant difference was found neither in calcification here nor in respiration for origin or pH treatment. Only trend was observed with a decrease of calcification in with a low pH. Here we have ambient pH and here a low pH, and for both sites. Thus, under the experimental condition, the colonies are tolerant to low pH regardless of their origin. However, in the CO2 vents, the adult colonies were smaller and had an encrusting morphology. They seemed to be weaker. Usually, corals' early life stages are more affected by acidification and warming than adult corals. So we tested if larvae from an ambient pH site were impacted by low pH and temperature. We obtained the larvae from ambient female colonies. Here you can see a larvae popping off the mouth of a female polyp. And we exposed them during 9 months in the lab with 2 different pH, low and ambient, and 2 temperature, ambient and warm. The larvae, we observed the development of the larvae, and the larvae which were exposed to a warm temperature presented a quick metamorphosis, whereas the one that were exposed to low pH and an ambient temperature had a slower metamorphosis. Here we can say that temperature could decrease the larval dispersal by increases the metamorphosis time. We then monitored the development of the larvae by observing the settlement and the mortality. And what we can see here is that double stress, a warm temperature and low pH, decreases the settlement rate and also increases the mortality. When larvae metamorphosed and settled, we measured the surface of the base of the juvenile polyps, the part that colonized the substrate and strengthened the settlement. And what we observed was that the base was smaller with a low pH. And this might be explained by the fact that under low pH condition, we observed an exposed skeleton at the base, and something that we haven't observed with an ambient pH. We also had the chance to observe budding of our recruits, the apparition of new polyps on the first polyp. And what we observed was that we didn't have any budding for the double stress condition. And we finally analyzed the skeleton by macrotomography, 3D scanning. And low pH had an impact on the volume of the skeleton, here in the dark color are the low pH, and also on the number of the septa that were lower under low pH. The septa being this wall structure in the coral skeleton. However, interestingly, the skeleton density here was higher under low pH and ambient temperature. And this is something strange about this species. Dr. Teixeira observed the same trend in colonies living in the CO2 vent. They had a denser skeleton under the low pH of the CO2 vent, which is something really different from other studies where low pH mostly have a decrease of density of the skeleton. So early life stages of an ambient site are impacted by low pH, as we could see in this experiment. And as reproduction was also occurring in the vent site, we were wondering if early life stages from CO2 vent were tolerant to low pH. We sampled larvae from the vent and ambient pH site, and we exposed them for 5 months under an ambient pH, low pH of 7.7 and an extreme low pH of 7.5, which is a projection in the CO2 vent for the end of the century. This is exactly the same experiment as we've done with the adults, with the first experiment I explained. So first of all, we obtained a significantly smaller length for the larvae from the vent, here in red. And this might be due to the female of the CO2 vent not employing as much energy in reproduction because of a low pH stress, which is extremely energy consuming. And when we observe the development of our larvae, we see that settlement was lower for the larvae from the CO2 vent under low pH and extreme low pH. And also we had regardless of the pH treatment in the lab, a higher mortality from the larvae from the CO2 vent. So this is possibly due to the lower reserve of energy allocated by the parents. And this impact on settlement and mortality might reflect a possible decrease in recruitment from the colonies of the vent. We then measured the surface of the base of the polyps, allowing to assess the growth rate of the extension on the substrate. And the vent recruits presented a higher growth rate than from the ambient site, especially the one that had an ambient pH in the lab that completely thrived. So recruits from the vent might have a higher growth rate to counteract the impact of acidification. And interestingly, Dr. Teixeira observed an increase of a higher density on the adults of this CO2 vent, and that might be linked also to a higher growth. So in order to compare the phenotypic and development results to genetic, we did differential gene expression by extracting the RNA of 4 polyps per condition. And in this PCA, as you can see, we have used a full transcript of the recruits, and we can clearly see a difference between the two origin. We can see all the red dot for the CO2 vent and the blue from the ambient site. And we also obtained 92% of the differential gene expression explained by the comparison between origin sites, and very little was explained by the pH treatment. So this might be explained by the fact that there is little gene expression between these two populations, as Asteroides calcularis is being a brooding species. The larvae have a short pelagic duration, and also the vent site is a semi-enclosed cave. And a previous study of Dr. Teixeira has proven that these populations are genetically different too. Here, I present you the functionally enriched genes that are differentially expressed between the two origins. Here we can see that the few genes found are downregulated on recruits from ambient compared to the vent site. They are coding for catabolic enzyme, binding enzyme, genetic regulation, and cytoskeleton regulation. And for this last, the cytoskeleton regulation, this might be the beginning of an explanation of why recruits from the vent might have a higher growth rate. In adult colonies, the same was observed for genes that can be linked to calcification. This might explain the higher density on adults observed in the CO2 vent, and maybe a beginning of a proof of adaptation of the colonies at the CO2 vent. So along these experiments during my PhD, I saw that adult colonies of Asteroides are tolerant to low pH on calcification and respiration, regardless of their origin. However, alone or by synergy, warming and acidification have an impact on recruitment of growth of early life stages from ambient pH sites, and previous parental exposure to CO2 vent have an impact on the size, settlement, and survival of the larvae. And even if the parents are tolerant to low pH, early life stages will present a decrease in recruitment and post-settlement growth, threatening the future of the population under future acidification. And focusing on the CO2 vent population, given our results and with the studied parameters, the situ vent site of Ischia do not seem to favor some acclimatization for Asteroides calicularis. We have larvae that are weaker, smaller, less survival and settlement. Recruits have a lower rate of survival, lower budding, and the adult colonies seem to be weaker too. However, we have these higher growth for the recruits and also the higher density of skeleton on the adults. And so for the analysis, testing adaptation needs to be done to explain this possible higher calcification rates and density on the CO2 population. So thank you very much for your attention, and feel free to ask the question period, and you can contact me at this address if you have more questions, or if you're interested in my profile for postdoctoral positions. You can find more information too in the two articles here. Thank you very much.
Okay, we'll probably adjust the screen later on. Thank you so much, Chloe, for this very interesting presentation that I'm sure took a lot of work on your part, but I think it's maybe a base for the future research. And I'm sure there's going to be a lot of questions later on. Okay, so let's introduce now our second speaker, Maximiliano Szkope. He was affiliated with the University of Malaga in Spain. She's an environmental scientist and biotech that attended University of Malaga. His research is focused on aquatic system ecology and climate change responses of crustaceans and the other marine organisms. Techniques that he regularly uses in his lab work are flow cytometry, epifluorescence microscopy, and enzymatic activity. Maxi will be presenting basically a project analyzing the changes in mitochondrial abundance and size in crabs and mussels due to their exposure to future predictions related to pH and temperature. It's actually a project that Maxi has worked with me on at the National Institute of Biology in Slovenia. And so this is the first stage of our results and his results. And so it's a pleasure to meet him here virtually again and to say hello.
Hi, hello, welcome everybody. Thanks for attending. And now, can you see my screen? Okay, let's go for the first. Okay, hello everybody, and welcome to my presentation, Novel Approaches for Stress Detection and Quantification in Marine Calcifiers, the multiple climate change stressors that impact fundamental subsoil stress response.
Maxi, I'm sorry.
Yes.
Maybe you can put it in a presenter's mode. We still see it as the slides.
Sorry. Let me stop screening and share my screen. On my screen. Tell me if it's okay now.
Yeah.
Now it's okay?
Perfect. Great. Yes. Thanks so much.
So, hi, everybody, and welcome again to my presentation, a novel approach for stress detection and quantification in marine calcifiers, the multiple climate change stressors that impact fundamental subcellular stress response. Abiotic stress impacts on the organism in resulting in subcellular and physiological changes. Prior to observable physiological changes, a series of subcellular changes can occur in the cell. Such change involve the modification of protein production, enzymatic activity variations, organelle disposition, and also metabolic energetic disturbance. Everything is connected by the mitochondria because the role of mitochondria is essential, regulating the energy demand and supply of the organism, being this the reason to focus on. To understand mechanistically how the multiple stressors of subcellular effects are reflected on the organismal level, we work with multiple organisms, tissues, and representative classes of cells within. And also we use two techniques that represent the acute response versus the immune system response, and also the recovery period to understand the importance of the cell recovery after the stress period. For this project, we established some hypotheses and objectives. The three hypotheses that we established tell us that different tissues have differential stressor-specific response, with the combined response determining the overall organismal response. The second hypothesis is recovery is stressor-specific and will impact overall organismal recovery. And then the acute and delayed system response affects the energetic balance and the cell abundance respectively. While the objective of the project is, one, determine which stressor has the most detrimental effects versus to which the organisms have the capacity for a positive response. Second, elucidate the benefits of the recovery stage and how to predict organismal response from the subcellular mitochondria-related observations. For the experimental designs, we use a setup of 9 big mixing tanks where we could establish the temperature, the pH, the light, and also the oxygen. From each tank, a pipe conducts the water to 5 little tanks where the organisms were exposed. In this experiment, you use 2 different species. One is the blue mussel, Mytilus galloprovincialis, and the Mediterranean shore green crab, Carcinus estuari. The first step in the experimental setup was the exposure of the organisms to the conditions. This started with an acclimatization period of 5 days, setting up a pH of 8.1 for both species, but 17 degrees Celsius for mussels and 8.1 Celsius for crabs. After that, the condition of exposure for the mussels were of 7.2 pH, 7.5 pH, 23 degrees Celsius, and a combination of 23 degrees plus 7.5 pH. And that took 3 months, while for the crabs, the duration of the experiment was of 15 days, with low oxygen, pH 7.2, pH 7.5, 23 degrees, and the combination of 23 degrees plus temperature. Once finished the exposure, we start the recovery stage, taking 15 days for the mussels and 5 days for the crabs. After the exposure of the animals to the new conditions, the organisms were anesthetized, sacrificed, and dissected to take the desired tissues. After that, the tissues were preserved in a 4% PFA, PBS, plus a 10% of saccharose. Then, the tissues were homogenated in a mortar, and these were passed through a 30 micrometer mesh to remove the big piece of tissue. Flow cytometry was used to measure the cell abundance and fluorescence. The cells groups in the flow cytometer were determined by their position in a forward size scatter plot, grouping the group cells in polygons. After that, the polygons, the number of events in each polygon was measured to determine the amount of cells in each category. In order to measure the changes in the mitochondrial fluorescence, MitoView was a cellular dye designed to attach the mitochondrial protein and emit green light when blue-green radiation is applied. The fluorescence was recorded in the green fluorescence forward scatter plot, using quadrants to establish the position of the control and compare it with the stress organisms, getting the difference of switching the cells from one quadrant to another. After collecting the data from the cytometer, this was subjected to a statistical analysis to determine the normality of the data and the distribution of the cells groups and fluorescence, which allows us to interpret the results. First, talking about the cells crab abundance, the crabs show significantly positive response in abundant cells to pH 7.2 across all the tissues, hepatopancreas, hemolymph, and gills. Also, in the hepatopancreas, we saw an effect of the combination of the pH plus temperature, and in the gills to the pH of to the 7.5 pH. These are the 3 major stressors with positive response, showing capacity to cope, following the same trend in granulocytes than in gelinocytes. On the other hand, temperature either destroys the number of granulocytes in the hemolymph or does not show a significant increase in hemolymph or in gills, indicating that temperature can be detrimental to the organisms. Interestingly, there is no response to low dissolved oxygen, which might mean that there is, well, or there is no sensitivity, or there is no capacity to respond to it. Once in, once in the recovery, while the organism could not respond to temperature under the stress, we see an increase in the cells under 28 degrees. Sorry, one moment. Here. Under degrees. The stress response at 7.2 and 7.5 stayed high despite the recovery, which also is not good, as it could have negative energetic implications. While pH 7.2 affects immediately to the organisms, the pH 7.5 needs more time to generate the negative effect. On the other hand, pH plus temperature stress goes down in the recovery stage, meaning the organisms can reduce the level of stress. Even during the recovery, the low dissolved oxygen does not induce a positive response, indicating a very negative effect of the low oxygen on crabs across all the tissues. Well, once about the fluorescence of the cell, the comparison with the fluorescence tells us the same story, that pH 7.2, pH 7.5, and the combination of pH plus temperature are the major stressors, because the fluorescence increase in the same treatments, following the helinocytes, the same trend, but with lower values than the percentage of fluorescence. The same indication is not increased under high temperature, confirms that this stressor is one that the organisms cannot cope with, hence they do not respond to, showing no extra fluorescence on the cell analysis. However, more subtle response in the granulocytes of the hepatopancreas tell us that low dissolved oxygen is a stressor that they try to respond to, but again, cannot mount a significant increase either in helinocytes or hepatopancreas. So the capacity to lead the organisms being the most impacted by the stress is confirmed by the mortality estimates. Low dissolved oxygen causes the greatest mortality, while increased temperature also have some impact. Interestingly, low pH of 7.2 also have some higher than expected mortality, which can be explained by the continuous response with increased cells that is energetically expensive. So the capacity to not respond to leads the organisms being the most impacted by this stress. Now, if we go with muscles, the muscles show significant positive response in abundant cells to pH of 7.2 and 7.5 and increased temperature in hemolymph, but not anywhere else. This means that only hemolymph as an organ is positively responding to stress through the increasing of cell production. On the other hand, other tissues show pretty much non-significant response, indicating that there is no coping mechanism to multiple stressors. This could mean that the organism is not well adjusted to the stress even after 3 months of exposure. Although the organism after 3 months of exposure show no positive response, as indicating of a coping mechanism. The recovery stage strongly indicates the increase in cells, meaning that the organism once the stress is missing and they have more favorable conditions, they can recover from the stress. The fluorescence of the muscles indicate us that the cell production across the hepatopancreas and the gills is steady, and that does increase. So the subcellular activity indicates an increase of the mitochondrial activity as an increase of the fluorescence, showing that cells are mounting positive response in terms of either increasing mitochondrial number or increasing mitochondrial activity. This likely means that while the organism have enough energy for increased subcellular activity, they likely do not have enough energy for cellular immune function, possibly meaning that the organisms do not have the best coping mechanism after 3 months of the stress. But what do the subcellular markers tell us? Basically, under the stress response on the y-axis, the organisms can alleviate the parameter, could be the cell number or the fluorescence, can reduce the response, have a negative response, or the response stays the same. With a positive response, this means the activation in the organism of the coping mechanisms. So the organism has capacity to respond to the stressor. In the recovery, if the response is on a high level, this means that the organism is still stressed, but if the response is on a level close to the control, this means that the organism is recovering or is recovered. If we find a negative response, this means that the organism do not have a capacity to respond. The only option is to look if the recovery can help such organism. So, with the recovery, we'll look all the three possible outcomes: a positive one, an indifferent one, or a negative one. If a negative one occurs, then the recovery doesn't benefit. If no response occurs, it means that either there is no sensitivity to the stressor. If the response is positive, this means that the organism can benefit from the recovery. We can compare the same responses, the no sensitivity response, with fluorescence to get more detail about the response. This is especially beneficial when we are not able to detect in the abundant cells to a specific stressor. So, what does this mean? No sensitivity, it means that there's no capacity to respond. So, now it's time to examine the fluorescence response and compare it to the response of the abundance with no significant change. If there is an increase of the fluorescence, then we have a positive response. If there is no change, then it's likely there is no sensitivity to the stressor. In short, crabs after 15 days of exposure have the capacity to increase the response across most all of the tissues and stressors, with expectation of temperature and low dissolved oxygen, which represent two stressors they will likely have the most problem coping. And then the recovery after a short time exposure, 15 days in crabs as opposed to 19 days in mussels, leads to a different response compared to the long term, with less pathway activated in short term, likely because the primary response is still high. And if we go with mussels, the mussels, even after 3 months of exposure to low pH of 10.2, can mount a positive response, high mitochondrial activity as well as the cell abundance. However, such high activity is present throughout the recovery stage, indicating a huge energetic expense, which is very likely unsustainable. Mussels, after 3 months exposure, barely show any coping response to all stressors, apart from ethylene, which means that all three stressors can have a detrimental effect unless the organism is subjected to the recovery stage. And in conclusion, we can affirm that stressors differentially impact subcellular, cellular, and tissue functions. It's a complex response on the organismal level, but this provides more mechanistic understanding of organismal physiology. The use of flow cytometry in combination with the method of staining opens very different interpretation of the response, coping mechanisms, and the importance of the recovery. The duration of the stress impacts the recovery and the intensity of the response. And these are preliminary results with n = 3 sample size. The variability is likely masking more explicit response, but this requires additional analysis. Thank you so much for your attention and hope you enjoyed.
Great. Thank you so much, Maxi, for this wonderful presentation. And if you guys, Chloe and Maxi, are ready, we should proceed with the questions. And I will be starting with the questions from the box here. All right. So Nadine Schubert says that this was a very interesting presentation, Chloe, and she's wondering if the lower recruitment under low pH is supported by field data of lower abundance or cover of corals at the vent site.
Yes, thank you, Nadine. Dr. Nuria Teixeira, in her article of Global Change in 2020, have done some monitoring of the population, and she hasn't seen any difference of coverage between the CO2 vent site and ambient site. She's now analyzing image is photograph of long-term long-term photographies, and she have seen that there is a higher mortality of the recruits in the CO2 vents. However, we don't have any information about the initial recruitment. That's it.
Okay. And then we have another question for Chloe from Samson Job. And it is, what was the reason, or were the reasons for the observed high shell density of the studied organisms found in the vents?
We don't actually have the reason of the higher density of the skeleton of the corals. A possible answer is this higher transcription of genes from calcification in the adults and in the juveniles. However, we don't have yet the reason. The fact that they are more dense might be a defense, facing the low pH to protect the skeleton. If it's denser, might be less impacted by low pH. However, we don't have the real reason about this high density.
Okay, great. Thank you. Anyone else? Any more questions? Couldn't find it in the box. So I'm going to use this time to ask Chloe still, and this is a little bit, following Nadine's question on if really adults are, pretty much resilient to these conditions, but the recruits are not. How do you have a sustained population of the organisms at this one site? Did you see the gradients in their distribution in the field? And mostly, what are your next steps in regards to this work? Is this something that goes directly into population demographics model, or what do you think are important next steps to close the gaps that we don't know about prospective survival of this species under projected changes?
Yeah. To answer your first question, these species are long-lived species, and we know that, we have almost an idea about the starting of the bubbling. Might have started 20 to 50 years ago. So the colonies that are present in the CO2 vent site might be from this period where the CO2 events were not active. And also we have seen reproduction in this cave. So there is recruitment, even if it's lower. We have seen recruitment, we have seen growth of the recruits. And as we have seen larvae settling just next to the adults, we know that the larvae are not, or very few of them are going out of the cave. We've seen that the colonies are not as big and bright as the one that we have in the ambient site. So in the future, they might decline, that's for sure. But yes, the next step would be to do more genetics on these species to see if they are well adapted or not, and if they are just going to decline.
Mm-hmm. Okay, so it's kind of like a natural selection already in place at this vent site, and maybe the Asteroides is more of a loser in these projected changes, and some other corals that might have adjusted mechanisms to cope better with the stressors, maybe they could prevail. Do you think your results are more extrapolated across species, or it's really a question of experimental results to be able to determine that?
Well, this score is quite special because it's an internal brooder. So the larvae is not traveling a lot. For other species, for example, Cladocora cespitosa, that I had the chance to study during my PhD, is a massive spawner. So he will just release the gametes in the water, and they will be drawn by currents, fertilize, and then the larvae will swim until they mature and swim. There is more chance to have a renewal of some population with this kind of species than with Asteroides calicularis, which larvae are already mature to settle.
Okay, great, yes. So it's really a specific strategies that different species are deploying that will determine their vulnerability or maybe their resilience, it seems. Yes, great, thank you so much. The other panelist has a question for Chloe, asking how long have the volcanic area been releasing CO2 bubbles, and over what temporal scales should we address adaptations? Is this happening over the centuries, decades, centuries, or millennia?
At CO2 vents, it's around 20 years. The bubbling has might have started around 20 years or 50 years around the Castello Aragonese, if I'm not mistaken. And the adaptation to the vent site is not, then it's not a question of centuries, more decades. However, as this species might have been in this cave for longer, and as they have this particular way of reproduction, maybe this population was already special.
Okay, good.
Okay, thank you for your question, well, for your response.
Here we go. Okay, so I haven't seen any questions for Maxi in this, so let me ask a few questions in there. So Maxi, why have you been using exactly those kind of species, and how did you determine on the exposure conditions, the duration times, and maybe if you could tell us a little bit about your now general feeling about the recovery time and the importance of inclusion of the recovery time in the experimental conditions. You're muted.
Sorry. Okay, by first we use the blue mussel as a Bible, because it's a huge distribution range with a lot of conditions from the Eastern Mediterranean to the Western Mediterranean, with a huge Atlantic influence, so more oceanic conditions, to the West Mediterranean with more tropical and the Red Sea influence. So it's a bivalve that it can habitat a wide range of conditions. So it's important to determine which one are the conditions of stopping the population or are affecting in a major way. And for the crab, this is a Mediterranean species that is becoming an invasive species all around the world. So it's important to know where are its limits to the distribution. Nina, can you please go back and to the middle of the question, please?
The importance of the recovery, of the recovery time in experimental conditions. Yeah.
Okay, once the organism is stressed, we can observe and see how it's going inside the cell. But also, once the damage is finished, this is still harming from inside. So we need to determine how long it takes to recover, completely recover, or if it's going to recover the organism after that impact. And above all, is the two keys is the strength of the impact is like the difference from the normal conditions we are going to be exposed, and how long is going to be exposed. So that even if we put the organism in recovery, it's not going to be enough for the organism to survive, because the cellular damage is so high that that animal is not going to be able to survive anymore.
Okay, great. I do have one more question, because I noticed that you two both use very different techniques in your observations. And I know Maxi will have more results on the enzymatic activities and so on. And Chloe has pretty much used such a variety of different techniques across different levels of biological organizations. And so my question is for both of you to discuss maybe, what do you think out of the methods you have used, which one of them can be applied for the in situ monitoring, and in which developmental stage these techniques are, how much more improvement they need, so somebody can just take basically the method you have developed or applied with pretty much sure results and then try to determine the same results in situ.
Okay, I think that, for example, the technique that Chloe used, the transcriptomics, it is very easy to take a sample. You can have your animal alive, just take a small piece of tissue, frozen, and go to the lab. And it could be a good way to determine which genomic sequence are active or not, to determine in which state of adaptation is that species. For example, if it presents, like she said, more genes related to the septa generation or to the expand of the cover of the skeleton, it could be an indicator that that population is going good, is having no problem with coping with the new condition. But if, for example, if it presents an extra production of some genes determined to the adaptation to that kind of stress, it could inform us that we're having problems in our community before the physiological response appears, and just without a minimal invasive analysis for the animal.
Okay, would you like to add? Yes.
No, well, I'm a little bit confused about the question. I think all the
yeah, maybe just to clarify, if you have done, basically you've taken the organisms from these gradients and put it in the experimental conditions, but how do you think, which of your techniques you have employed would be most useful if you just wanted to get like a response from the natural environment, where maybe the conditions are not so clean, confounding parameters, stuff like this, exposure time and stuff like this?
Yeah, as Maxi was telling, I can just freeze some samples and take them, but also we can do some of the measurements I did in the lab can be done also in situ without damaging the colonies. For example, respiration or calcification with, for example, the alkalinity anomaly technique with samples of water after putting them in an closed environment, and also photographs for the growth, for example. Almost everything can be done in situ. And I think also what did Maxi can also be done. Also, they think, well, you have to find your animal and kill it, but all the experiments can be put in situ and leave them there in a naturally acidified environment, for example.
Yeah. Okay, I was more thinking about, like, out of so many techniques, what is preferential technique that you might have a recommendation to use for easier recognition of how the coral is doing in the field or not?
Oh, yeah. Okay.
Okay, good. Thanks so much. Okay, so we have also a hand from the audience, or the question from the audience. Maria Cristina Gambi, please.
Yeah, good evening, and compliments to Chloe Carbonne. I would like just to clarify, since I work in the cave long ago, much longer, I want to say that the first data on asteroids in the Grotta del Mago date back to 1976, when just few colonies were present in the cave. Why? Because at that time, the distribution of Astroides, which is a thermophilus Mediterranean species, was very strongly limited by the temperature. So Ischia was at the northern limit of the distribution of this species. Northern than Ischia, the species was no more present along the Italian coast, at least in the Tyrrhenian coast. Then just these few colonies, when we did the survey for the marine park, the Ischia Marine Park Institution in 2000, the population was very, very much, it was increasing a lot. The cave was almost covered by Astroides. I want to specify that venting was present even in the 76, but was very, very, very limited, was very, very weak bubbling. And again was very quick, very low bubbling even in 2000. Then we sample again in the cave for other reason. In 2014, if I remember, and then we found the bubbling that was incredibly stronger and increased a lot, but still the distribution of the Astroides was at the same level of the 2000. So I think that the population started to increase in the cave due to global warming of the area. Now Astroides is present up to the Tuscan archipelago, which is more than 300 kilometers north of Naples, of the Gulf of Naples, north of Ischia. So global warming has favored the distribution and even the abundance of this coral. And bubbling, I think, is something like 15, probably 15, something around 15 years stronger than before, which was still was present, but was very weak. We measured it in 2000 also the pH, but we couldn't see any difference respect to ambient. On the contrary, in 2014, the pH was definitely lower than the ambient. I hope just to clarify a bit the history of this very interesting site. Thank you.
Grazie. Grazie mille, wow. Maria Cristina Gambi. Yes, all this information of the history of the place, I didn't really know, but you are the expert of this place. Thank you.
You're welcome.
Yeah, thanks, Cristina. I think this long-term perspective is definitely something that we want to know more about and integrate in a biological understanding, because it really clock sticks on the long term for biology. But since we have a pretty substantial community here online, I just wanted to really inquire and ask if anyone here of the community working in the Mediterranean maybe is working with some other coral species or crabs and mussels that both Chloe and Maxi presented, and either experimentally or in the field, and what are their results? Maybe just fast comments on that to see how much variability we have across biological responses and what sort of environment people are working in. So if anyone wants to take a stab, maybe.
For the corals that I've been working on, I think we are the only one that had natural populations in a naturally acidified environment. Mm-hmm. At least we are the only one working on naturally acidified population. So a lot of work has been done on these species with pH, temperature, And they have seen that they were quite robust, the adult colonies. However, we are the first one working on the early life stages.
Okay. Interesting.
And also with the global warming that was Maria Cristina Gabi was talking about, these two species of corals are suffering a lot during the marine heat waves during summer. Yeah, a lot of necrosis for asteroides and bleaching for Cladocora, and it's worse every summer.
And when you have like a recovery event or after the marine heat wave, do you observe that organisms are quickly recovering as well, or is that time dependent, or do you have any insights on the recovery as well?
In situ, there is a lot of work done. I've done some experimental experiments, and the only real suffering they have suffered in the lab was plus 3.2 degrees the whole time of the experiment, following the natural evolution of temperature. Cladocora bleached and asteroides necrosed, and we've seen some recovery of color for Cladocora and regrowth of some polyp with asteroides. However, I think that in situ it's worse than in my experiment.
Okay, great. Good to know. Okay, so we also have a raised hand from Nuria. So go ahead, Nuria.
Yeah, can you hear me?
Yes, we can hear you.
Okay. Thank you for organizing very nice talks. And Maximiliano, nice to meet you. And just, what I would like to add with this dialogue that we are having is that, also you mentioned about natural populations, how do they respond to ocean acidification? And maybe just to add some words that Chloe, she has already said that there are other natural populations of corals in the CO2 vent systems in Panarea, for example, that there is balanophilia. Yeah? And also, if I recall well, it has been described a new natural population of Cladocora near Sicily. And these systems, they are very useful as a natural analogs, and also knowing what it's meaning to work on natural analogs. And maybe also just to try to synthesize some very nice points that we have, that also some people they have asked, and curiosities, is that maybe there are some general patterns of the response of calcifying species, and more precisely, corals, but also there are some specific species response, like, for example, asteroids. Why asteroids, in order to cope with this stressful, environment that it's mean with low pH, why, to calcify more? But at the same time, the colonies, they have lower polyps, and they are smaller. Then maybe this is a strategy, in order to cope, in face to this stress. Just, wanted to say maybe hello, and to add these very few things, And this is true that it has been in Ischia, the vents of Ischia, they are dynamic, and we are working, very close with geology, and the venting activity, it has increased over the last 20 years. And this is what we are, trying to answer, no? What are the mechanisms that allow this species, no, to grow, to survive in these sites? And maybe also one thing to add is that maybe the population, they are there, and they can live, they survive, and they grow, but maybe they thrive in these conditions. That maybe what we think is that they are present, but they are continuously, under stress. And this is also one thing that we are, trying to reveal. And that's all. Thank you very much again for this seminar and this afternoon. And okay, thank you.
Great. Well, thank you, Nuria, for this really interesting insights. I was thinking that maybe working with geologists is really crucial, because if we could go back 30, 40 years and try to get some sediments out of these corals, we could maybe compare how corals were responding under less venting as well. So maybe there is a more material that can be collected, and obviously this could help us understand if this 20+ years of increased venting is enough to spark some adaptation for these corals to survive. So I think these are very interesting questions, and obviously interdisciplinarity here is the key. All right, let me see if there are any more questions, but otherwise, I think on our side, on my side, I would like to thank Chloe and Maxi for wonderful presentations and giving us insights on what's happening along the Mediterranean coast in different regimes. Maxi work in the Northern Adriatic, and Chloe obviously of this vent site. So great insights to multiple stressors, and I hope the story continues and we understand this system so much better in the future. Thanks so much to everybody and the organizers.
Thank you so much for coming and for listening to us. It was a pleasure.
Yes, thank you very much to everyone for the attending and the organizers. Thank you, Nina, for the presentations.
Absolutely. Thank you, guys.
Bye-bye.